Okay, so these are our karyotype notes for the day and how the day is going to look here. So I have some quick notes we'll give on what karyotypes are, and then we'll do independent practice very quickly, just to pull some of those big ideas of the notes and give you some practice. We'll, of course, go over it so you can check your understanding. We'll do another quick round of practice, and then that should be it for the day, and hopefully we're feeling pretty comfortable with karyotypes by the end of it. Remember, you can always email me if you happen to have any questions, and I'll do my best to get back to you as soon as I can. Okay, so up to this point, we know that humans have a total of 46 chromosomes in their body cells, or 23 pairs of chromosomes when we're organizing them by homologous pairs. So the one that you received from your father's gametes, and then the one that you received from your mother's gametes. So on the left here, I have a picture of a karyotype. So what a karyotype is a picture taken this in a little bit here. So a picture taken of chromosomes during cell division to test for genetic disorders. So a picture taken of chromosomes during cell division to test for genetic disorders. So now, kind of what happens here, so the reason why it's taken during cell division, that's the only time when chromosomes are visible. So remember, before cell division, DNA is all loosey-goosey in the nucleus in the form of chromatin. And so it's not when cell division happens, so when we start to undergo mitosis, where the chromatin condenses into packages that we call chromosomes. And generally, this picture, while it could be technically taken at any point in mitosis, that's our division for our body cells, it's generally taken during metaphase, when the chromosomes are all lined up in the center of the cell. So that way you can clearly see them one by one. So that picture is taken, and then there's kind of like a cutting and pasting that goes on with the pictures. And so whoever is analyzing, creating the karyotype, organizes it in a particular way to make it easier to identify any abnormalities, so anything that could possibly be wrong with the karyotype. Now, if you notice, the chromosomes, they're arranged from biggest to smallest. We end with the shortest chromosomes at the end. And they're also numbered here 1 through 22, and then we have the sex chromosomes at the end. So these chromosomes 1 through 22, these have a special name that we call the autosomes. And they're arranged from the largest pair to the smallest pair. So these are your chromosomes that code for all of a variety of different types of heredity information. Maybe on chromosome number 1 is hair color, and on chromosome 11 is height. But what makes these different from the sex chromosomes is the fact that they're more so controlling everything about your body aside from what sex you'll be at birth. The sex chromosomes are at the end here, and it's the sex chromosomes that determine those characteristics that a particular sex will have. And so that's what kind of keeps them a little bit separate from those autosomes, as well as what sex that you're born with. Females have what we call XX for their sex chromosomes. So if you receive 2 X chromosomes, then your sex is female. The X chromosomes happen to be fairly large when compared to our other option, which is a Y chromosome. So a male has one X chromosome and one Y chromosome. So these sex chromosomes, these are put at the very end as the 23rd pair. So it's these sex chromosomes here that are the 23rd pair. All right, so here we have this little empty dot here, so we're going to throw a sentence in there. So abnormalities in karyotypes. So abnormality meaning something that is not right, something that's different, abnormal. So abnormalities found in karyotypes can determine genetic disorders. So what I mean by abnormality, so what we are looking for is something that is not expected in a normal human karyotype. So we're expected to have 23 pairs of chromosomes, 46 total, 22 pairs of autosomes or 44 autosomes, and then 2 sex chromosomes. So an abnormality would be maybe I have an extra 9 chromosome, or maybe I'm missing a 9 chromosome. So an abnormality would be having more or less chromosomes than what you're expected to have. And we've talked a little bit about this in meiosis, so non-disjunction, so those chromosomes don't separate correctly. And so because of this, you could have one extra or one less. So we call one extra chromosome a trisomy. And so it'll be named by the extra pair. So a trisomy 20 would be 3 of chromosome 20 instead of 2. And if we have one less, so just one chromosome total, so maybe only one of chromosome 22, we call that a monosomy. Now, as we move on, we look at inheritance by males and females due to their sex chromosomes. It's kind of important to understand what's going on there, to understand like how sex is determined during reproduction. So meiosis helps to create those gametes, and we talked about how we have sperm that are found in males and eggs that are found in females. Now, since females have 2 X's as their sex chromosomes, during meiosis, so the making of gametes, it doesn't matter how many cells are made, all of their gametes are going to contain an X chromosome. So half will get that first X that was received from mom, and half will get the second X that was received from dad. But there's only X as an option. So all of egg cells will have an X chromosome. Since males are XY in the making of gametes in meiosis, where we're creating those haploid cells, that have half the number of chromosomes, half of the sperm that are made will have an X chromosome, and the other half will carry a Y chromosome. So because females can only offer an X, but males can offer an X or a Y, it is the male that determines the sex of an offspring. So it's a male that determines the sex of a baby. And so that's why, too, you have a 50% chance of being of the male sex, and then 50% chance female. Is just the probability of, is it a sperm with an X, meaning the egg with an X, or is it a sperm with the Y, meaning an egg with an X? Now, we've talked a little bit about non-disjunction before, but just since it's been a while, we're going to throw this vocab term back down on our notes since we're going to talk about it again. So it's non-disjunction that causes these karyotype abnormalities, or not necessarily the karyotype abnormalities, but just the abnormalities in the number of chromosomes that we can identify on a karyotype. So remember, non-disjunction is an error in meiosis where homologous chromosomes fail to separate correctly. So during that anaphase period where they're in metaphase and then in anaphase they split apart and separate. So if there's not an even separation, we could end up having one cell receiving more than one chromosome or one cell receiving less than they're supposed to receive, or maybe just a portion, maybe it like separates in half. All right, so this is it for the front side of the notes here. So at this point in time, we're going to pause the video and we're going to take a second to work through this back page. So this should probably take no more than about 10 minutes. If everybody gets done sooner than that, then go ahead and start. So take about 10 minutes here, go through your notes, do your best to answer some questions about these karyotypes, and then we'll go through it. So video's not paused yet. Take a second to pause it, please. Okay, so at this point in time, everyone should have worked through our first practice with a karyotype on the back of our notes. So we're going to go through it real quick just to check our answers, check our responses. So this picture on the right-hand side here, so this is called a karyotype. So we've got our chromosomes arranged from largest to shortest. There's total pairs of chromosomes, 23. So we have the 22 autosomes plus the pair of sex chromosomes. So 23 pairs total. 22 are autosomes paired. Total autosomes, so all autosomes that you see, 1, 2, 3, 4, etc., would be 22 plus 22, or 44 total. There's 2 sex chromosomes. And so if we're determining what the sex is, so I have 1 chromosome over X, 1 over Y. So this would be a male. Now we're going to use the karyotype below to answer some questions. So down here, there's nothing over the Y, but I have 2 over the X. So this person here is biologically female. And I know this due to the 2 X chromosomes. Here you had to identify some likenesses, so some similarities that you see among paired chromosomes. So the first is in general, they're about the same, like height and size. So I don't have this one partnered with this one down here. So they're about the same height and size. You may have also noticed too that I'm just going to use the words like same coloration, discoloration here, but if you look very closely at the 2 pairs, at the partners here, so you'll notice that in a lot of these areas where it's darker are the same, and areas where it's lighter are the same. So these two white patches match up here. These two white patches match up here. These two white patches match up here. These two match up here. So during the staining of the chromosomes to make them more visible, since they're homologous or they're the same, you're going to see kind of like that same coloration between the two chromosomes. So that's another likeness, another similarity that they have. So if you have maybe two that are kind of like close in size, so maybe like these two here, and you are trying to see, okay, which one should they be partnered with? You know, this one here is pretty solid black, whereas this has these two noticeable white gaps. So you would be looking for a partner that would also have those two noticeable white gaps, not one that would be just solid black here. Our trend in size, so as we go from 1 to 22, they do overall become a little bit smaller, a little shorter. All right, and then our last one here, so you had to describe a noticeable difference between the X and Y chromosome. So the Y chromosome just happens to be much smaller in size than the X chromosome. And then here you also had to identify a genetic disorder. So Down syndrome is a genetic disorder that's caused by the presence of all or parts of a third copy of a specific chromosome. So some of you might have done this with your non-disjunction slide, Down syndrome. So maybe you remembered it from memory, but applying our what we're looking for with karyotypes here. So looking at the karyotypes, we're looking for which number happens to have 3 chromosomes. So all of these have 2 until we get to 21. 21, there's 3 chromosomes there. So it's chromosome number 21 in the autosomes that is the cause for Down syndrome. And it's also called a trisomy 21. So trisomy meaning 3. Okay. So at this point in time, I would say we're going to leave these notes out. And we have one more practice that we're going to take the time to do here. So it's called Karyotype Worksheet up at the top. And so we're going to take a second to, once again, pause the video. We're going to get this handed out. And we'll give everyone, again, should take maybe about 10 minutes or so to work through it. If we get done sooner, then great, go ahead and start. But if not, depending on how everything's are going, if we need another minute or two, that's fine. But we're going to take a second to pause the video, and then we'll go through this worksheet, the answers, once everyone is finished. Okay, so at this point, we should have worked through the karyotype worksheet, part 2. And here you're going to do a lot more diagnosing. We do have a few questions, though, first, before we start. So you're asked here, how many total chromosomes are found in a normal karyotype? So 46 total chromosomes are found in a normal human karyotype. And we're expecting to have 23 pairs. Again, chromosome arrangement goes from largest to smallest. We have the sex chromosomes at the end. And how do we determine sex? So what are we looking for? So you're told to try to be as specific as possible. So we're looking for we're looking at pair number 23 at the very end. If I see 2 X chromosomes, then this is a female. If I see 1 X and 1 Y, then this is a male that I'm looking at. Now you have a table here to help you to identify a variety of different types of common genetic disorders. So you're going to use this table here as you go to help you to identify what type of genetic disorder it is that we are looking at, if we even have one. Like this first one here, this is a picture of a normal human karyotype. So I have 22 pairs of autosomes. None of them have any extras. None of them are missing anything. And then I have 2 sex chromosomes, 2 X's. So I have 46 chromosomes total here. This person is female with the 2 X's. And this is considered a normal karyotype because I don't have any abnormalities, no trisomies, so no extra chromosomes, and no monosomies where I have 1 less chromosome. And this next one here, so I do happen to have an extra chromosome. If I go through and count, I have 47 chromosomes total. And the extra one is a trisomy, happens to be here at 18. This person is male, 1X, 1Y. This would be an abnormal karyotype though. I have 1 extra chromosome. So to figure out the syndrome, so the extra is at 18. So I want to see where is chromosome number 18 affected. So here's where I'm expecting to have a trisomy 3 of 18. So this is Edwards syndrome. Those of you that didn't do Down syndrome, so you'll see your possible karyotypes popping up here as well. So on the back here, so we have an abnormality. So chromosome number 47, or not 47, oh my goodness, I have 47 chromosomes. This person is female. I have 2 X's here. So since I have 47, this is an abnormal karyotype. I have an extra at chromosome number 13. And so at chromosome number 13, this would be Patau syndrome. Karyotype number 4, so we do have an extra chromosome. I have 1 X and 2 Ys. So since I do have here an X and a Y chromosome, the sex of this person would be considered male. It is, though, abnormal, so this man is supposed to only have 1 Y chromosome. Instead, he has 2. And so this is disorder XYY. It doesn't have a fancy name. Instead, it's just XYY syndrome. So extra Y chromosome. All right, and then lastly here we have Down syndrome, so trisomy of 21. This person here is male, 1 over X, 1 over Y. So this would be an abnormal karyotype because we have 47 chromosomes instead of the expected 46. All right, so hopefully we feel a little bit better about karyotypes today. Again, if you have any pressing questions, please feel free to send me an email. I'm also going to put an Amoeba Sisters in Google Classroom too, so that way we can watch that. If we have any possible questions, and that video might be able to clarify some things for you and make them a little bit easier to understand. So hope you all have a nice day, and we'll hear from the substitute, so what to do next.